Welcome to Biology with Alyssa Turcott and Patricia Amori. I am one of your hosts, Alyssa, and today we'll be talking about gene cloning. But first, I will let our other host introduce herself.
Hello, everyone that's tuning into our podcast today. My name is Patricia, and I'm so excited to tell you guys about gene cloning.
I am also so excited to tell you all about gene cloning, so let's get started. First and foremost, we have to identify what gene cloning is. And gene cloning is a process in which a gene of interest is located and copied, or in other words, cloned out of all the DNA extracted from an organism. This process is going to help make multiple copies of the same DNA molecule. Now I'm going to hand it over to Patricia so she can go through the steps of gene cloning. Go ahead, Patricia.
Thank you, Alyssa. Now, with gene cloning, there are 6 steps to go through to actually clone the specific gene. The first step of gene cloning is that DNA is going to be extracted from an organism.
The second step is when the bacterial plasmids are cut.
The third step is going to be when the gene's DNA and the cut plasmids are combined into a test tube.
The fourth step is the recombinant plasmids are transferred into bacteria using heat shock.
For step 5, the bacteria is plated out and is allowed to grow in colonies.
And lastly, the gene library is screened to identify the colony containing the gene of interest. Now that we went over the steps, I'll let Patricia have the spotlight.
Thank you, Alyssa. Now, since we know what gene cloning is and the steps we have to take to get there, we should probably go into the history of gene cloning. I've done a lot of research about the history of gene cloning, and I would love to tell you all about it. So, the history of gene cloning starts with 2 men, Herbert W. Boyer and Stanley N. Cohen. In 1972, Boyer realized that the enzyme EcoRI cut DNA and left staggered ends. Previous to this discovery, when scientists needed to cut DNA, the DNA would be cut clean. And they would have to make molecular additions so that the DNA could be joined with other pieces of DNA. But with the realization of EcoRI and its ability to cut DNA and leave staggered ends, scientists no longer had to make molecular additions and could just cut and go with their experiments. Both Cohen and Boyer came to a conference in 1972 to give papers on a meeting on plasmids. Plasmids are DNA that's found in bacteria and is physically separated from chromosomal DNA. Usually, in most cells, DNA is found within the nucleus, but in bacteria, it's spread out throughout the cytoplasm. Boyer was studying the nature of DNA ends produced by the EcoRI cuts, and Cohen was studying a procedure that allowed bacteria to intake plasmid DNA to create offspring with self-replicating plasmids. This basically created clones. They both started a collaboration over dinner one night, sending research between their labs and sharing findings. Cohen would isolate plasmids at Stanford and then send them to Boyer. Boyer would then cut them with EcoRI and analyze the DNA fragments. Cohen would then join them with E. coli, the plasmids then multiplying. There are certain plasmids that can make bacteria drug resistant, which was the basis of their next experiment. And this experiment started with the plasmid PSC-101, which was cut by EcoRI on only one site, and this left the plasmid's ability to replicate intact. The plasmid was then joined with other DNA and introduced to calcium chloride-treated bacteria. This mixture was then spread on a culture containing tetracycline. Tetracycline is a medication that treats bacterial infections by killing the bacteria. The bacteria with the artificially copied DNA survived the tetracycline, meaning that Cohen and Boyer had grown drug-resistant bacteria. They continued their experiments and later proved that genetic materials could be transferred between species. They wanted to capitalize on their findings and founded a company called Genentech. They realized that they could probably clone human genetic info, and this means that humans' means of fighting disease could be copied, and this could possibly remedy birth disorders. They both succeeded in cloning somatostatin, a hormone produced in the brain that regulates growth, which is the copied version was virtually identical to the naturally occurring substance. They both constructed a plasmid coded for human insulin, which was approved by the FDA in 1982 and became the first biotech product to appear on the market. For their research, Boyer received the Biotechnology Heritage Award in 2000 for his development in rDNA tech. He also won the Perkin Medal in 2007, the Winthrop Sears Medal in 2005, and then Cohen won the 2016 Biotechnology Heritage Award for his work in creating rDNA plasmids. Is there anything you would like to add or ask me about?
I found it very interesting that they grew drug-resistant bacteria, um, and how Boyer learned how to create clones. So I found what you said was very interesting.
Thank you, thank you. The research definitely hit close to home. My best friend in the whole world, he's type 1 diabetic. And so Boyer and Cohen developed artificial insulin, which my best friend needs to survive. He wouldn't be able to go on without it. There's just not enough naturally occurring insulin that humans could farm for the amount that it's needed. And so this is life changing research that they were doing.
Yeah, and it's really great that they found out all these things so they could help, people like your friend. Um, but now that we know what gene cloning is and its history, Patricia, have you ever wondered what the purpose of gene cloning is?
I know from my research that there's a lot of medical reasons for gene cloning, such as the insulin thing. Um, but outside of medical reasons, I'm not quite sure.
That's true that there is a lot of medical reasons for this, but what I found is that one reason is to gain information about the nucleotide sequence of the gene, and this reason can be used to study a particular gene or even compare versions of a particular gene from a different source. And for the second reason, for gene cloning, is to manipulate a particular gene, and that is simply means to alter a gene's DNA sequence. Manipulating a gene can also be used for combining different DNA molecules together. And I have another question for you. Why do you think manipulating a gene would be a goal for gene cloning?
Um, Manipulating a gene could possibly go back to, again, back with the medical purposes, it could possibly have reasons for like remedying birth defects and things like that, and just furthering DNA studies.
That's probably really true. I'm sure that they use it for medical reasons, but for what I found is that the reason for manipulating genes is to place the modified gene into the cells of a new organism. And with all that being said, what do you think about the information that I went over?
I think you had some really great information. Like, yes, we do have all this information and all this technology, and it's a really great thing to have, but how can we use it? It was really, really interesting to see the different ways the scientists use this technology. Thank you.
Yes, I also found it very interesting, and what you found out was also I learned a lot more about gene cloning through the history and what you found out. Um, but that is all we have on gene cloning.
I hope you all enjoyed tuning in to our podcast, and hopefully, you learned something new or found our podcast just entertaining.
Until the next episode.
I'll see you then.